In 1945, babies were born in the Netherlands during a famine so severe their mothers were starving. Decades later, those babies, now adults, were at much higher risk for diseases like diabetes and obesity. Something had changed. The famine was over. Food was back, but these babies were still storing fat like the famine was ongoing. Scientists checked their DNA. It matched their parents, no mutations. So why were their bodies still in survival mode?
Genes aren't like light switches. They're more like doors. Some wide open, others sealed shut. Whether a gene is expressed depends on how easily RNA polymerase, the enzyme that copies DNA into mRNA, can get in and start reading the sequences and go on to complete transcription.
DNA wraps around proteins called histones in order to stay compact. In this form, it's called the nucleosome. But chemical tags like methyl and acetyl groups control how tightly it's packed. When it's loosened, RNA polymerase can get in and read the gene. But when it's tight, the gene stays unread and unexpressed. It's the same DNA code, but different instructions because of these chemical tags. These chemical tags can survive cell division and even be passed across generations. They affect gene expression without changing the DNA itself. This is epigenetics, the hidden machinery behind inherited trauma. Those Dutch babies inherited DNA from their parents, but the genes controlling metabolism were still tagged with famine signals. Even decades after the famine ended, their bodies kept storing fat, just in case food went out again. In a world with plenty of food, that message backfired, raising their risk for diabetes, obesity, and even cancer. But this isn't the end of the story. Scientists are finding ways to remove harmful epigenetic tags, treating diseases like cancer, and restoring healthy gene expression. Our DNA remembers, but with new tools it may learn to reset.